Hi guys, so this video is going to clarify the gap between transcription and translation. Maybe you want to know more of what exactly happens in the process leading up to the beginning of translation, because so much of what happens during the post-transcriptional processing are so critical in that if something goes wrong or there's a mistake in the processing, that mistake could lead to a number of different things, including dysfunctional proteins or a protein that is not intended to be synthesized. And that's not a good thing at all. And by the way, if you have missed the transcription and the translation videos, I'll leave the YouTube cards for you on the screen. But before we jump into the actual post-transcriptional processing, I think we should recap just a little bit. So in transcription, recall that the DNA strands are being uncoiled by the enzyme called RNA polymerase. That will move down the DNA so it creates an RNA. Remember, the whole purpose of transcription is the synthesis of an RNA. There are different types of RNA that could be transcribed, including tRNA, which is transfer RNA, messenger RNA, or mRNA, and rRNA, also known as ribosomal RNA. And for our particular example, we are transcribing or making a messenger RNA. The first aspect of post-transcriptional processing is the addition of a chemical group called the cap. This is the cap, CAP. This cap is going to bind to the 5' end of the messenger RNA. This messenger RNA is actually the pre-messenger RNA. RNA. And you find out why it is in a second. Now, what does the cap have to bind to the messenger RNA anyway? Why can't this messenger RNA leave the nucleus without this cap attached to it? The job of the cap is to control the initiation of translation of this messenger RNA. So what happens is there will be initiation factors in the cytosol where translation occurs that will recognize this cap on this messenger RNA, and they are going to bind onto the cap, which then will allow this messenger RNA to be translated. On the other side, the 3' end of the messenger RNA, there's a change of chemical basis called poly-A tail. This poly-A tail will attach itself to the 3' end of the messenger RNA. This poly-A tail is made of multiple adenosine monophosphates. So you have this nitrogenous basis, adenosine. You have a repeating of those bases. It's basically a stretch of the messenger RNA that has only adenosine bases. What it does is it's going to protect this messenger RNA from enzymatic degradation in the cytoplasm. Remember, we're not in translation yet, so this messenger RNA is still in the nucleus. So this component of the messenger RNA, the poly-A tail, is very important to be attached to this pre-messenger RNA. In addition, it increases the stability of this messenger RNA, and it's been shown in studies that it can play key regulatory roles in enhancing translation efficiency. It helps with the termination of transcription. Once the cap and the poly-A tail binds to the messenger RNA, the messenger RNA will go through a process called RNA splicing. During the making of the messenger RNA, the RNA polymerase is building what is known as the pre-messenger RNA. So then, what is pre-messenger RNA? Now we understand that a gene is a requirement to make amino acids, and those amino acids are used to build proteins. Well, messenger RNA is the carrier of the functional genetic codes for a gene, and within a gene are regions of excess bases called introns. As you can see here, the white regions here are the introns. So, introns. These introns do not code for the amino acid sequence of proteins. However, they are transcribed in the initial messenger RNA molecule, that is, pre-messenger RNA. But they must be removed before the mature messenger RNA leaves the nucleus. So these introns are going to be cut out of this pre-messenger RNA molecule. The remaining segment of this pre-messenger RNA, more precisely known as exons. These are the exons. They are the regions of the gene that are actually functional, in which they have the ability to produce amino acids. So these introns are going to be cut out from this pre-messenger RNA, and these exons are going to be joining each other to become a mature messenger RNA. So here we have several different segments of exons, right? So one here, 1, 2, 3, 4. So we have 5 different segments of exons. And these exons, what's interesting here is that these exons can be spliced in different ways. So that means that, this segment of exons doesn't have to be 1, 2, 3, 4, 5. They don't have to line up in that way. They could be 1, 3, 2, 5, 4, or 5, 4, 3, 1, 2. So with each splicing, it leads to a different code, and therefore the synthesis of a different polypeptide. Okay, you can imagine that when the introns are cut out, these segments of exon can be arranged in different ways. And through this mechanism, one gene can code for several proteins. Now, once the RNA splices is completed, this pre-messenger RNA becomes mature, and it's ready to move through the nuclear pores and enter the cytoplasm, where, of course, the translation occurs. This is the mature messenger